A French and Indonesian scientific expedition has explored a largely unknown region of the island of Papua New Guinea. The researchers ventured into one of the planet's richest areas of biodiversity, the Lengguru Mountains. For the scientists, the territory is like an open-air laboratory. It was formed 11 million years ago when the Australian and Pacific tectonic plates collided. The relief which soared up raised uncrossable barricades, bristling with peaks, beset by abrupt cliffs, dug out by chasms. The animals and plants that lived there were trapped. To survive, they had no choice but to adapt very quickly to the new conditions. How did the species that are prisoners of the mountains acclimatize? Have they developed new functions? What has changed in their feeding, their metabolism, their morphology? The expedition had some intriguing encounters. A forest kangaroo, a cricket that emits surprisingly powerful ultrasounds, and a large pigeon from the family of the legendary Dodo, extinct for centuries. The samples taken from the specimens discovered at Langaro have been transmitted to cutting-edge research laboratories. They will help science to understand the evolution of the living world and foresee the future of our planet in a period of intense and chaotic change. There is no land access to the Lengguru Mountains. To reach them, you have to pass by the sea. French biologist Lengguru has roamed Papua New Guinea for more than 15 years. During a previous expedition, he saw the extraordinary biodiversity of the Lengguru Mountains.
Mana bos? Masih berapa lama sampai Lubu?
3 jam kita sudah tiba di Laut.
Berarti masuk sebelum malam.
Ya.
Lengguru has convinced Gono Symbiotic, biologist at the Indonesian Science Institute, to take part in the adventure. Together, they share the direction of a mixed team, including Indonesian and European researchers.
Okay.
The Lengguru Mountains are a little like a giant natural maze. It's an enormous limestone mass, twice as large as Corsica for its land part. It starts at the sea bottom in the abyssal trench of Seram, at least 2,000 meters in depth, and it rises above the sea to 1,500 meters. So the interest of this expedition is to understand how all this living architecture has been put in place over millions of years and to predict its future evolution, faced with the great challenges of global warming and human activity.
Lobo is the only village at the foot of the Lengguru Mountains. That's where the team of scientists disembarks. The expedition base camp sets up close to some dwellings. The arrival of strangers is an event for the villagers. For the men, it's an opportunity to make money. The expedition can't get by without auxiliaries, the only ones capable of carrying equipment in difficult areas.
Your name?
Justuf.
Mamal 2 orang. Mamal itu maksudnya mamalia. Ada yang tentang kelelawar. Orang gajah ini gabung. Berarti moluska.
Everyone checks the condition of the equipment they'll carry one last time. The heaviest loads are split up so they can be carried on a man's back in difficult conditions. It's the last night the scientists will spend together before splitting up to go to their research zones. During 8 weeks, the mammal, bird, insect, and plant specialists will use all their expertise to discover the secrets of the mountains. According to the villagers, a species of kangaroo lives in the dense and bushy undergrowth of Lengguru. This is surprising information, as kangaroos are usually a plains animal. Gono Semiyadi sets off after the tracks of the mysterious marsupial.
I really looking now for kangaroo footprints. We are hoping we can get some indication of the presence of the kangaroo coming to the water. Oh, looks like this is the last night footprint. You can tell from the moist of the footprint.
In this kind of undergrowth, it's difficult to follow such a tenuous track for a long distance. But close to the river, Gono Semiyadi has found a place where there are more tracks than elsewhere. Up till now, the marsupial has never been photographed or filmed in its natural environment. Without leaving a trap, the only way to proceed is to install cameras that trigger automatically and can also film at night.
But the angle, this is about the right angle. Is up to about 2-meter coverage.
Now, 2 or 3 days must pass, counting on luck. The cameras were in the right places.
Yang ini kayaknya nggak ada garis punggung.
Tidak. Lebih ke daerah utara New Guinea.
Yang ini kayaknya ini pendulum testis.
Iya, itu yang gantung itu kayaknya.
The recorded film offers a surprise. This very strange animal doesn't jump as the plains kangaroos do habitually. It moves around by leaning constantly on its tail as if it were a fifth leg. The Lengguru kangaroo seems to be different from the most common species of marsupial. This discovery intrigues the scientists. Zoologist Geraldine Veron is a mammal specialist at the menagerie of the Natural History Museum of Paris.
It's quite an intriguing, interesting animal. I don't quite know what it is. It seems to me to be a land animal. You can see it moving around on the ground. It doesn't jump like a wallaby either. We'll have to have a look at what its means of locomotion is. Is it an animal which stays on the ground or does it climb trees?
A comparison with the species present in the menagerie may give some of the answer.
Well, these are Bennett wallabies. So wallabies are marsupials, animals where the young develop in a pouch. They live mainly in Australia, in Australasia in fact. And here we can see that they're animals which are called macropods, that is with large feet and with a leg which is used for jumping. Well, it acts a little like a spring. It's in a Z shape. And they also have little upper paws, the hands, which aren't used for movement, they're mainly for catching food. They live in open spaces like prairies. And here are Dendrologus Goodfellow, which are tree-dwelling kangaroos, also marsupials, macropods like the wallabies, but which live in trees. So they're adapted for this. They have a very long tail, which acts as a balancing pole. The front paws are very developed, and in particular, they have very big claws, which let them grip tree trunks. And the back paws are smaller than in a kangaroo or a wallaby because they don't jump as much. It's an animal which lives at the top of trees.
Neither a plains wallaby or a tree kangaroo, the Lengguru marsupial poses a few questions. Geraldine now turns to the museum's historical collections and its stuffed specimens.
Right, we're looking for small kangaroos. We'll see if we can find it. Ah, it might be that. Yes, you see? These little forest wallabies. Yes, if you're looking at New Guinea, that matches well. I'll get it out. It's like the one you saw. What would be really interesting would be to see if Anthony can tell us something about the morphology of the paws in relation to tree dwellers, to see if it's purely a land animal.
Dorcopsis hagini was discovered in 1897 on the island of Papua New Guinea. Up until now, no one has studied its skeletal structure.
So here we have the X-ray image. That's the paw here, with the tibia descending. There are the phalanges here, and what really interests us to better understand the adaptations to a tree or land environment is the posterior part here. This is called the calcaneum or heel bone. The heel bone is interesting because in there is inserted a tendon, the Achilles tendon, connected to the largest muscle, the gastrocnemius. This muscle is extremely important because it serves the extension of the paw, and that gives it most of its force for movement. For this animal here, the length is quite different from what I've seen on typical plains kangaroos from Australia. Large macropods which travel great distances, so that suggests there's something very special about these animals.
Yes, that's interesting.
I have some images of tree kangaroos. Let's look at them.
Oh, yes, yes.
So this is the same X-ray but of a tree kangaroo. Here it's the other way around, so the paws are this way and the claws, which are very well developed, are here. And you can see that the heel bone is a lot shorter, and so this animal can't jump as far or run as fast as the animal we've just seen.
The kangaroo skeleton from Langaro has bone characteristics which are intermediary between a tree kangaroo and a plains kangaroo. Under the pressure of the environment in which it was isolated, the Papua New Guinea marsupial evolved, and during its exile, it became a new species which could be called forest-dwelling. Its evolution shows the formidable resilience of living beings which managed to adapt to profound changes in their environment. However, the Lengguru kangaroo is under threat.
Terus sekarang kalau sudah gini mau dimakan. Iya. Saya suka.
As they do everywhere in forested zones, the local population eat meat from the bush, and the little marsupial is easy to catch. The scientists are confronted with the old dilemma they meet everywhere where strong protection measures haven't been taken to safeguard fauna in danger.
So setiap datang pasti dapat?
Iya, tergantung
juga.
Tergantung rezeki.
Tergantung rezeki. Sekarang rezeki lagi bagus.
Iya.
Tapi Bapak tidak pasang untuk yang pakai tali jerat itu, untuk rusa gitu tidak? Pasang juga?
Ada pasang juga tetap di sana.
Di sana?
Iya.
Terus berapa hari sekali Bapak jerat itu Bapak periksa?
2 hari.
2 hari. Kali pasang berapa jerat?
Biasa 5.
Hanya 5?
Sampai 10.
Hanya 10? Dalam 2 hari pasti dapat?
Bisa.
Oke, silakan Pak.
Preserving the kangaroos requires the active participation of the inhabitants. Once they are made aware and trained, they become the best caretakers for the site.
Ya, sudah Pak.
The flora of Papua New Guinea includes around 1,300 tree species and 15,000 species of plants, most of which exist nowhere else. Because of this, it is one of the most diversified and rich areas on Earth. Botanists Vincent Droissat and Jean-Christophe Pantot from the Institute of Development Research methodically gather plants which they think show unexpected characteristics.
At the moment, things are very good. We've collected more than 50 flowering plant species and nearly, I'd say, 200 or 300 non-flowering species. We've collected a lot of living specimens that we'll then try to cultivate in Jakarta, and then another part here in Papua New Guinea.
At the edge of the sea, the forests of Langaro are regularly flooded by the tides. Swollen with salt, the soil is poor in nutrients. To subsist, plants have developed original strategies. A large number of plants grow on top of other species. These species are called epiphytes, or air plants. Plants that feed on insects are very diverse, and there are many in this specific environment.
Toutes ces stratégies de vie qui n'utilisent pas le sol.
All the growing strategies that don't use soil are flourishing in this environment. Here there's a plant which captures small prey in these urns, where the insects come and drown. For example, in this urn, there are 4 or 5 big dead ants, and their decomposition will help feed the plant.
Contribuer à l'alimentation de la plante.
Other plants don't eat the insects, but prefer to use them.
Can we pull that off to perhaps see? Because the entrances for the ants seem to be under the rhizome. There's one entrance. 2, you can see the structure in this stalk, which has been modified, which is full of cavities, in fact, where the ants can live. I'll use my secateur, it's simpler. This is the first time that we've seen a fern which associates with ants in this way.
This fern is a myrmecophile. It lives in symbiosis with an ant colony. The insects make their nest in these hollow protuberances. In exchange for the shelter, they bring the fern the mineral and vegetable nutrients that it needs. Samples of the plant discovered at Lengguru have been sent to Germinal Rouen, fern specialist at the Natural History Museum of Paris, to be identified.
So, let's see this specimen.
C'est un spécimen qui me plaît particulièrement.
I particularly like this specimen because it doesn't really correspond to the idea we have of a fern with a very split leaf. Here there are simple entire leaves. This particular stem, sprawling, we call the rhizome. And then what we can see swelling on the rhizome are these little black structures with a translucent membrane around them, which are what we call scales. So the scales, they're found not just on fish, but also on ferns and all parts of the fern plant. The scales are primordial for the determination of what these plants are. So we have to compare the size, the shape, the number, and the localization of these special scale structures. All of these characteristics allow me to determine that the plant is a Lechanopteris sinuosa. Lechanopteris are wonderful plants because it's quite rare amongst ferns to find ones that have modified structures to welcome, if you can say that, insects.
This astonishing symbiotic system is the result of coevolution, during which the plant and the ant have constantly adapted to each other. The symbiosis is such that without the ants, the plant would die, and without the plant, the insects would have no shelter and would not survive long. Some of the most complex and creative scenarios of the theory of evolution that exist are to be found at Langaro. The researchers couldn't dream of a more fertile land. Here, the night swarms with life. All the nocturnal species hunt.
We're going to need a bag.
For the herpetologists, the expedition keeps its promise. Pythons are quite common in the region, especially in Australia and Indonesia. But this is the first time that a specimen has been found at Langaro.
It's absolutely magnificent.
This is an amethyst python. Its name comes from the milky rainbow sheen of its scales.
It's not even very nasty.
Just watch out, it'll roll around you.
I've got my machete.
Trying to bite you.
That could work.
Is it the snake that smells like that?
Oh, yes. When they're not happy, they do that. I hope it doesn't burst the bag.
Alongside the sea crocodile, the python, which can grow up to 5 meters long, is one of the most powerful and formidable predators of Langaro. In the undergrowth, the researchers are listening for unusual sounds. That sound there, for example, we haven't heard it before. It's surely a frog which belongs to this type of habitat. We absolutely have to find it. We're not really used to working in this type of forest, searching in this kind of place.
There it is.
Oh yes, that's it. So it's a species we haven't yet seen. It's probably not been described yet. This is an oreophrene, and it croaks once or twice every 2 or 3 minutes, so it's very difficult to find.
What's impressive is the size of the beast in relation to the noise it can make.
Let me see the frog you've caught there. Great. It's a mixofixa. Oh, wow. I don't know what this thing is, Philippe.
Me neither.
Oh, it's even two-toned, did you see? There's one part. What's that thing? Those tubers on the eyelid. This is a super discovery. That's great.
Not bad, eh? It's got attitude. Personally, I love it. It'll jump on me next.
Tony Robillard is an entomologist at the Natural History Museum of Paris. Insects, and especially crickets, fascinate him strongly.
So here we have a female Rhaphidophora. In fact, they have asymmetric antennae. There's one antenna which is a lot longer than the other one, and they often move them in a kind of circle around themselves, doubtless to find out what's around them, to see if there's a predator or an obstacle. There are very few things like these creatures in any case, with this behavior. Yes, there it is. I'm putting the microphone not too close and not too far away either. I don't know this at all, in fact. It might be a different species. I can't say for the moment, but from the sound, there's something different. So it's really interesting.
To try and identify this curious species of cricket, Tony Robillard dissects 2 individuals which he conserves in ethanol.
These specimens have a very particular kind of face. This kind of white mask which covers all the cricket's face, that is without a doubt something new. This can be confirmed by looking at the male genitalia. They're very characteristic of cricket species. We make an incision here along the abdomen. And we can see the genitalia. This part here is very unusual. I've never seen shapes like that.
Voilà les genitalia.
Ending in a kind of spatula like that. It's really very strange. It's something which is completely unique in crickets. So there I can say that we probably have a new genus.
Discovering a new genus is an exceptional event in the life of a researcher. The genus is an entire line of a species, up until now unknown, and presenting the same characteristics. This discovery is an important advance in our knowledge of the living world. The Natural History Museum of Paris has one of the best collections of insects in the world. Tony Robillard pursues his investigations here, in particular on the song of the crickets of Langaro.
So the song is made up of units, which are in fact syllables, and that corresponds to this sound here. That's what is used to construct the architecture of the song. So this syllable here, we can disassemble it and look in detail at the sound wave. And you can see that it has a frequency of 10.5 kilohertz, which is twice as high as that made by an ordinary cricket. This is a very important characteristic, exceptional for this species found at Langaro.
The jungle resounds constantly to the noises of birds, frogs, and insects. To be instantly recognizable by its partners, the cricket of Langaro has developed an original technique. Just like bats and dolphins, they emit ultrasounds which pass through the surrounding cacophony.
So, to understand how the male produces this high-frequency sound, I used a camera which records at 3,000 images per second. And that allows me then to measure in slow motion the speed of wing displacement during production of the song. Each time that the wings close, a syllable is produced. And we can already say that it's a movement which is, despite the slow motion, very rapid, which allows the cricket to produce very high frequencies. It's something you can't see at all with the naked eye, and it's a lot more rapid than for an ordinary cricket. This is a new system of communication using high-frequency signals, so it's really original, something completely new. Now we're going to have to look at the other end of the system, with the females, to see where the signal is received.
How do the female crickets of Langaro manage to capture the ultrasounds emitted by the males? Tony Robillard goes to the University of Leipzig to meet the neurologist Stefan Schornich, who has developed a unique experiment in fundamental research.
How are you?
Very good.
Good flight?
Yeah, it's nice to see you again.
Okay. And actually, I just have one recording running here.
Yeah.
And so I inserted some electrodes in the cricket, the connection up to the brain, and I can switch a loudspeaker on, so you can actually hear the activity in the central nervous system. Let's start with some sound we can hear. So with not a very high frequency in our hearing range. So you can hear the sound pulse, but you also can hear the activity of the neurons. Now let's see for the really high frequency, which we cannot hear. So this is 20 kilohertz now, and I present the sound pulses in the same pattern. And the only thing we hear is the response of the neurons. So the cricket can hear the 20 kilohertz. So they have a really high frequency hearing.
The brain of the female cricket reacts to the ultrasounds. This would mean it can perceive vibrations that other animals can't hear.
As a
neuroethologist, I try to understand how animal nervous systems can affect behavior. To put it in another way, I observe what, in the case of an evolution in behavior, happens in the brain. And I'm specialized in the acoustic communication of crickets. Working with Tony is fascinating because we have the same interest for crickets, but two different areas of expertise. When we bring our knowledge together, that is the genealogical study of Tony's and my study of the nervous system, that allows us to progress towards a new understanding of the evolution of the species.
To confirm his hypothesis, Stefan Schoeneich looks at the minuscule brain of the insect, and more precisely, the nerve cells specific to its auditory system.
So I just put the preparation in the microscope.
Okay.
So the microscope is now scanning through the brain level by level. And what we know from the normal field crickets is that this round area here, this like a ring, this is the area where the auditory information is processed. And what you see here, this branch which goes out here and goes all the way up here, this is very typical for the high-frequency neurons.
Okay, so that's confirming that they lost low-frequency perception, finally.
Yeah.
And that's really cool, because it's indicating us how evolution can generate a new system of communication by grabbing some neurons and giving them a new function. Yeah. So that's the only time we could prove these kind of things with such details.
Yeah, I guess so, yeah.
Cerebral imaging shows that the females can only perceive ultrasounds. They can only hear male crickets of their species emitting on high frequencies. In a way, the Lengguru crickets communicate on another radio frequency than other common crickets. If they hadn't evolved in this way, the continual hubbub of the jungle would have prevented females and males from finding each other to reproduce. In the lost world of Langaro, changing to survive is a desperate necessity for many species.
France!
The team of ornithologists number the birds present in the mountains with the help of the villagers.
Yang ini. Ini ada. Ada di sini. Di atasnya ini ada. Ada.
Okay.
Namanya hutan ini semua gambar ini ada.
Yeah. How about this?
Ini.
Mambruk.
Ada.
One of the stars they hope to observe is the western crowned pigeon, or gura. It's an endemic species in the region, but its origins are still a mystery to science.
Everything in here, it's in around here, but you need time to catch them. Yeah, I know.
Christophe Thébault from the CNRS, Borja Mila of the Museum of Madrid, and Hidayat Ashari from the Indonesian Science Institute estimate that in a square kilometre of forest there are up to 150 species of bird. Such diversity is unique in the world.
The nice thing about this place is that we can actually get canopy species because we're very high, we're at the level of the canopy. So it's a very steep ridge on both sides. So birds that are flying through the canopy at this height, they're not gonna go up here. They'll just stay at this level and continue. So we have a good chance of catching things that otherwise in the understory we would not catch. Okay, very nice, beautiful. It's wine color and the golden underwings, beautiful. Tout à fait magnifique. Oh. Both. Not very happy.
Individuals of every colour and every size pass through the expert hands of the ornithologists. Before releasing the birds, the team examines the state of their plumage. They measure the wings, the beaks, and take a few drops of blood for genetic studies. For 2 days, Christophe Thébault and Bojamila have been tracking the crown gora.
Boja, I've just heard a gora. This is a very good habitat for them.
Yes, yes.
We'll see if we can find a feather or something.
Okay.
Look, a gora feather. It's a wing feather.
Like it doesn't have white tips, right? Very impressive. Very nice. That's an amazing crown.
This bird with an extravagant crest is one of the rare survivors of the family of large pigeons, which has continued to decline to the point where it is now feared that it may disappear very quickly. Its weight can reach 2.5 kilos, but its wings allow it to fly and perch in trees. It could be a relative of the now extinct dodo, a bird regretted by all scientists.
Would be nice to catch. Nice to catch. Perhaps with a feather.
To know if the crown gora of Lengguru is really a relative of the legendary dodo, reconstructing its genealogical tree has to be attempted. The first step consists of comparing the DNA of the species conserved at Langaro with those of other specimens of gora kept in collections around the world. The ornithologists go to the sequencing platform of INRA at the Genopole of Toulouse to consult a geneticist, Olivier Boucher.
Hello, Oliver. How are you?
Hello, I'm well. And you? Where are you back from now?
Oh, from a long journey. Papua New Guinea. We'd like to study a group of pigeons, very large pigeons, the largest in the world, which are called crown pigeons. And we'd like to understand where they're from, how they arrived, and when the different species were formed. The challenge for us is that the material we'll have to use is essentially museum specimens, which are 10 years, 50 years, even 150 years old.
Okay, so since they're museum specimens, we'll be working on degraded DNA.
What are the technical difficulties?
Well, because the DNA is degraded, that means the DNA is in little fragments. Not only in little fragments, but as well in small quantities. So what I propose is that we make some tests on some samples, see what that gives us, and if that's satisfying for you, we'll be able to sequence and study all your samples to answer your biological question.
Well, a very exciting aspect of this project is that it's an extremely international effort. We're using a lot of samples from for example, the Natural History Museum in Great Britain, in Australia, in Indonesia. This international component is very important to bring together all the resources and expertise to truly understand the diversification of gora from many different perspectives.
In Jakarta, Hidayat Ashari from the Indonesian Science Institute is in charge of taking the necessary samples to send them to the Toulouse Genopole. The gora feather found in the forest will give data about the individuals living now at Langaro. The gora is now a species that is protected by Indonesian laws and international conventions. Collections have very few specimens, all taken before the 1970s.
With the old specimen like this, that mostly they has arsenic in it, in the body and everywhere. We cannot use the part from the body. Mostly we use toothpaste. That it not a lot of arsenic around this area, so we can cut it, just piece of maybe around 2 millimeter.
While the sequencing of the DNA taken from the collection specimens is happening, Christophe Thébault and Borja Mila attack the next challenge. To establish the parentage of the gura, its genetic heritage has also to be compared to that of other large pigeons, notably the dodo. Endemic to Mauritius, the dodo was a bird that was almost a meter high with an average weight of 10 kilos. Its withered wings prevented it from flying and made it an easy prey. It became extinct at the end of the 17th century after Europeans landed in its environment. Lewis Carroll, in the 19th century, made the dodo immensely famous by making it into a sympathetic character in his book Alice in Wonderland. Since, the dodo has become an icon of the extinction of species which man is responsible for. At the Natural History Museum of London, the paleontologist Julian Hume has just discovered new bones of the legendary bird.
Hi, Julian.
Christophe. Great to see you.
Yeah, good to see you. It's been a long time.
It has been a long time. Hi, nice to meet you.
Nice to meet you. So what are you up to?
Well, painting dodos as usual. And talking of dodos and giant pigeon, tell me about your crown pigeon gura project.
The project is going very well. Now what we need to add in the story are the big extinct pigeons.
I have actually found some cave material which might help the cause.
Yeah.
As you can see, this is the famous Marasange material, always is chocolate brown, but unfortunately it's totally useless for extracting DNA. Just by luck, I found a new cave on Mauritius. There's a very obvious difference. This one's brown and this one's white. This is from a limestone cave. But the thing is, this is producing DNA because it's been in a cave, protected environment, and it's not suffering like this one would do in the elements of Mauritius. So hopefully this is the key.
Absolutely, absolutely.
Well, these are small pieces of bone. Do you actually want to see the complete skeleton?
Be great.
Great. Well, let's go over here.
So, Julian, I mean, the crowned pigeons are the largest pigeons in the world right now. So why have all the other big pigeons got extinct, you think?
Well, there's one key thing, and that is mammals. The dodo evolved on an island where there was no terrestrial mammals, whereas the crowned pigeon had a whole range of predatory mammals to have to contend with. Now, if you look at the size of the wings, they're about the size of a chicken on this huge bird. That is because flight is so expensive to maintain. So if you don't need flight, why have it? Why maintain it? And the dodo didn't need to fly. Everything it needed was on the ground.
So when the mammals were introduced onto Mauritius, so the dodo just couldn't cope with that.
There's a lot of tales about humans actually hunting dodos to extinction, but that wasn't the case. In fact, they introduced mammals, pigs, deer, goats, and so on, which competed with dodos for food, but the worst culprit was probably the black rat, which was accidentally introduced. They would have bred up into huge numbers, been a serious predator of dodo eggs and chicks, and that was probably the key reason why dodos became extinct and the crowned pigeons are still with us.
In terms of history, large pigeons are more vulnerable than other birds. It is urgent for the scientists to see if the gura does belong to that family, to construct a policy of preservation. Jade Bruxot, doctoral student in Christophe Thébault's team, takes samples of various species to the Paleogenetic Center of Copenhagen, one of the rare laboratories in the world which works on ancient DNA like that of mammoths or early man.
Hi, good to see you. Ooh, thank you. I got a suit for you. Let's see I hope it's the right size. So, how old are the samples?
Probably end of 16th century.
How many samples do you have?
There are 5 inside.
5? It's
Yes.
amazing.
Yeah.
Where did you get them from?
They come from the British Museum. Julian Hume sent us them.
I'm really looking forward to see these. Okay, ready?
Ready.
Perfect. Let's see what we can do with this.
Thank you.
Thank you.
Why did we come to Copenhagen? Because they have a lab specialized in ancient DNA here, which is very, very good. It's one of the most up-to-date laboratories in the world. Here they work on samples dating from the 1700s or even older, and they need sterile conditions which are as close as possible to perfect.
Et ici, on a les conditions adéquates pour faire ce travail-là.
Wow. So maybe we should just start with one sample first,
Yeah.
one at a time.
Okay.
But this is so much. I think we should break it into smaller pieces and extract it individually.
Okay.
Okay. It look very well preserved.
Hey guys.
Hi.
How's it going?
Hey Tom.
Well, I think.
Yeah? How far are you?
These are the libraries.
That's the libraries already?
Yep. So it's
Exciting.
ready for sequencing, I think.
It looked really promising.
Mm.
Well, I guess fingers crossed.
Yeah.
Yeah,
Forward.
exactly.
Let's go sequence.
Oh yeah.
So the really big challenge is because the animal has died and decomposed, not only is the tissue, the muscle gone, but the bone is broken down, and within the bone, the DNA is broken down. So a very simple analogy is, for example, comparing a whole book that I give you with a book that's been through a paper shredder and is in lots and lots of small pieces. We have to work with these tiny fragments. We have to sort out what is the real DNA and what is the bacterial DNA, and then we have to piece them together to try and understand the information that's present.
At this stage, the most developed technological tools and the best experts have been mobilized to create the genealogical tree of the Gora.
Hi.
Hi, Jade. All okay?
Yeah, fine.
Right, you've come to give us our results.
That's right. In the end, we managed to sequence 39 samples.
No sequencing problem? From the DNA you sent us, we had no problems, and we have very, very good sequencing results.
Up till now, we thought there may be 3, maybe 4 species based on color criteria, and now we can confirm there are 4.
4?
So that's quite interesting.
It's interesting and unexpected, in fact. Great. And now the relation to other large species of pigeons?
Well, when we compare all the large pigeons, they're all found together. Our crown pigeons are close to a pigeon with a strange beak, 2 others that look more like pheasants than pigeons, and also the dodo and the solitaire.
Okay, so that confirms a hypothesis that was already proposed and shows quite clearly that finally in this large group nowadays, the largest pigeon still living is the Gora.
That's it exactly.
This kind of result is really interesting and should, I hope, in any case, encourage the conservation of these birds and also the forests of New Guinea.
Absolutely.
That's perfect. Bravo.
This magnificent plumed bird is a survivor. While the mountains of Langaro are a refuge for the Gora, they're not as safe as all that. This large pigeon is a favorite prey for poachers. Draconian measures will have to be taken or it will disappear in its turn. The mountains of Langaro have kept their promise. It really is the Eden of biodiversity that the researchers felt it to be. However, it must not be thought that this natural citadel is invulnerable. Modern tourism has the means to get everywhere, and if mining companies discover wealth in the ground, the site of Langaro could quickly be sacked. To prevent what would be a tragedy, it needs to be turned into a sanctuary as fast as possible.